This video explains primer extension capture, or PEC, a technique that allows the targeted retrieval of DNA sequences from extremely low copy number, highly fragmented DNA sources. We have used this technique to sequence complete mitochondrial genomes from Neanderthal bones over 40,000 years old, and the method can be used for capture of any small target regions or single nucleotide polymorphisms, and from many individuals. The main steps in this procedure are PCR amplification of a primary DNA library, quantification of the amplified library by quantitative PCR, target primer extension on the amplified library, bead capture of primer extension products to remove non-target fragments, and elution and sequencing of the captured and washed targets.
Hello, I'm Adrian Briggs from Svante Pääbo's lab in the Department of Genetics at the Max-Planck Institute for Evolutionary Anthropology in Leipzig. Today we're going to show you a procedure for isolating specific DNA sequences of interest from a complex, highly degraded DNA source, such as ancient bones. We have used this procedure to study the mitochondrial genetic diversity of Homo neanderthalensis, or Neanderthal man. Let's get started.
The DNA used in our experiment comes from Neanderthal bones between 40,000 and 70,000 years old, which were found at 4 sites across the Neanderthal geographic range. A bone is first exposed overnight to UV light to decontaminate the surface of any extra